Hello, everybody. Welcome to a new episode of the Dissenter. I'm your host, as always, Ricardo Lopes, and today I'm joined by Dr. Peter Sterling. He is professor of neuroscience in the Perelman School of Medicine at the University of Pennsylvania. His broad goal has been to learn how the brain is designed to understand its functional architecture. He is the author of books like Principles of Neural Design, and the one we're going to focus the most on today, what is health, allostasis, and the evolution of human design. So, Dr. Sterling, welcome to the show. It's a pleasure to have you on.
Great, Ricardo. It's a pleasure to be here. I would just add that the Principles of Neural Design, it was a co-author. My co-author was Simon Laughlin, and he was very important. I don't want to leave him out.
Yeah, sure. I will leave that in the link to your books. I mean, it will be there anyway, so okay. For people to know. So first question then, how do you go about studying how the brain is designed?
Well, it's a big question, of course. It's really, I think, a sort of kind of reverse engineering. You have to accumulate a broad knowledge of what's there. You try to make a parts list, just for whatever little area you're studying. And then you have to construct some sort of a wiring diagram of how all the parts go together, a schematic, as you would for any machine or electronic device. And early on, I did this by what's now called connectomics. Connectomics is trying to find the synaptic connections between all the neurons in an area. And I did this working on the retina, the mammalian retina, starting in the 70s. And the way I did it, my lab did it for a long time, was to take a piece of retina, fix it, and slice it up into very, very thin slices, about a tenth of a micron thick, and then photograph each slice in the electron microscope, because you need that resolution to see synapses, and then to trace out. It's like cutting through a bowl of spaghetti and reconstructing all of the pieces of spaghetti as you go. And of course, we did it then with about 200 serial sections and a few thousands of electron micrographs by hand tracing on plastic sheets. Now people send it out, and they get it sliced up automatically and photographed automatically, and then computers do most of the tracing, and then you have to annotate it and so on. So and they called it connectomics. I called it, in the early days, microcircuitry, trying to reconstruct the microcircuitry. And we found it was very slow, and I didn't have much competition because people thought it was just too much trouble. And now it's become almost a routine thing, and people understand that it's a valuable approach. So if you get circuits and you see broad patterns, then you have to have some hypothesis about what's going on. And so, for example, I thought of this example. One of the things you notice is in the brain, all over the brain, is that the tracks that connect one area to another by wires called axons, actually the axon diameters vary greatly. Some are very thick, some are extremely thin as you can make a neural process, so a tenth of a micron. So if you have a tenth of a micron axon and you compare that to a 10 micron axon, the difference is 100-fold factor, and the difference in the volume of those two wires and the energy use is 10,000. So it makes a huge difference whether you use fine wires or thick wires. And I used to show students in the lab. I taught a course on brain structure. I said, look at this. This difference is huge, and it must have something very important to do with the excuse me, the design of the brain. And so what would that be? And of course, supercomputer, we're smarter than a supercomputer, but a supercomputer requires a whole room to encompass it, and it draws 20 megawatts of electricity. Our brain fits in essentially in a milk carton, a little over a liter, and it runs on the electricity required by a refrigerator light bulb, about 15 watts. So how can you compute with so efficiently in space and energy? And that has something to do with these this variation in the axons. The fine axons require 1/10,000th of the space and energy, but they conduct information slowly, fewer bits per second. If you need, so auditory fibers are the very largest ones, and they need to conduct information rapidly, many bits per second, and so it costs more. So one of the principles of design that emerges is that the brain, wherever it's possible, sends information as slowly as possible. And when it has to send information fast, it has to be use much thicker wires, and it's much more costly. And so that's one idea of how design emerges from looking at patterns and thinking. The general hypothesis that Simon Laughlin and I used in our book was that the brain is really designed for computational efficiency, and that's why we can do so much better than a supercomputer.
Mm-hmm.
That's the idea.
Yes. So, but since you can study the brain at different scales, from the nanoscopic to the microscopic and the macroscopic, how do you integrate all of those scales?
Well, yeah, another big question. Well, yeah, so let's deal with what nanoscopic is. Nanoscopic refers to 10 to the minus 9th meters. It's a length scale, and so when you're dealing with the parts of the brain tissue, biological tissue, that's nanoscopic. For example, a protein molecule, single protein molecule, is about 6 nanometers across, okay, to give you some idea. And so at that level, the sort of signaling and interactions go by chemistry. And the space between the synaptic space that joins two neurons at a synapse is about 20 nanometers. And the packet of neurotransmitter that one neuron releases onto another is about 30 nanometers. So that's a scale that has been highly studied. My laboratory worked on that to some degree. And if you compare that to a whole neuron, say a 60 micron neuron, the size is like, the difference is 100,000-fold. So we have to understand the brain, we have to understand function across a scale, a minimal scale of 100,000. And then, of course, when you get up to connections between neurons, you're up to the millimeter scale, and you're adding another factor of 1,000. So one of the points about the nanoscopic scale in chemistry is that chemistry runs faster when the molecules that have to interact are at high concentrations. And so you can do pretty good, reasonably fast chemistry with high concentrations. And you do that because at small volumes, only a few molecules are enough to make a high concentration. And so at a synapse, when the brain releases one packet of transmitter into the cleft, synaptic cleft, this tiny cleft, it only needs like 5,000 molecules of this small chemical to create a fast reaction. The other thing is that chemistry proceeds by diffusion. Normally, in this aqueous or intercellular fluid is largely water. And in diffusion, it turns out the physics in diffusion is relatively fast at very small distances. At a distance of 10 nanometers, it goes in microseconds. Things diffuse from one place to another in microseconds. But as the distance increases to, say, a millimeter, it's very slow. And so, so the brain needs to use these very small volumes, small distances to make things go fast. And then when you get to the level of a neuron and signaling from one neuron to another across millimeters, diffusion simply won't work. And so you need to, you have two alternatives. One is that if you need to signal long distances from the brain to the body or from one area of the brain to another, if you release a small packet into the bloodstream, the heart pumps and carries things relatively quickly over seconds. And I mean, if once you get an injection of something, you can feel it, you know, sort of a jolt in a few seconds. But even that, seconds for the brain is a pretty long time. It's slow. So if you need to, you know, really see, if you're driving, you need to see something quickly, you need to respond quickly. And oh, I should stop and say, that method of releasing a chemical into the bloodstream and sending it all over the brain is what hormones do. And of course, when you release a small packet into the whole volume of the blood, it's very dilute. And so you have to make receptors on neurons and cells in the body that bind very tightly so that they can grab onto just the slightest molecule, low concentration, that it arrives. And that further slows it down because strong binding means slow release. And so it's a slow process relatively. If you need fast binding and unbinding, you need to send signals electrically over long distances and then release high concentrations at the other end. And so that's what nerve wires, axons do. They use electrical signaling to send information over long distances, and then they convert it at the other end to these chemical processes. So, but the electrical signaling is extremely expensive, and because you're releasing, it's basically you're discharging a battery, and then you have to recharge the battery, and it's expensive. So, you know, that's, we've gone now from, in terms of signaling, from the nanoscopic up to the millimeter level, which is a factor of 1,000,000. Okay, and then you know, if you want to study actually how the neurons in an area communicate with each other, then you have to record really the electrical activity of thousands of neurons simultaneously, and either by optical imaging or by electronic means. And the progress that's been made in that over the last 10 years is immense. And that sort of technology came as I was leaving, closing my laboratory and going on to other things. But so you have to sort of gradually master all these different levels and think about it. So.
Yeah. So now another big question, what is allostasis? I mean, what is it exactly? What do you mean by it?
Sure. Excuse me. Allostasis, I mean, the short answer is allostasis is a word that I made up, okay, with Joseph Ayer, my colleague Joseph Ayer, in 1988. And I made it up, it's from the Greek, two Greek parts, allo and stasis, with the advice from a professor at Penn whose philosophy, studies Greek philosophy. And before I explain allostasis, the reason I coined it is as a contrast to the standard word in biology, homeostasis. Homeostasis you find in every biological journal and almost every article. And the idea is that the body is a self-correcting mechanism, that homeo means constant, stasis means constant, and homeo is this self-correcting mechanism. And the idea is that the body really has a collection of corrections like a thermostat. And if it has a set point for temperature, for blood glucose, for heart rate, for blood pressure. And when something causes a deviation from any of these things, the error is detected and then feeds back a correction to make it right. And this has been a standard idea. It was started by Claude Bernard in the 1700s, I guess 1800s. Walter Cannon was a famous professor of physiology at Harvard around the turn of the last century, studied physiological regulation, and he coined this term, actually, homeostasis, in the 1930s. And every medical school teaches in the first year course, at least Penn teaches in the first year course, that blood glucose rises, causes insulin to be secreted, causes glucose to be absorbed, and it's all automatically regulated. The problem is that when I started looking at this in the early 70s with Joseph Heyer, I realized it isn't really true. I mean, as our activities change during the day, from night to day, our physiology changes. And when we become, and it's not just, Cannon recognized the condition he would call fight or flight. If you're challenged in some way, there's a neural response. The brain recognizes this challenge and it mobilizes chemicals and increases your heart rate. And so you're prepared to meet this challenge. But his understanding, and still to this day, the understanding of regulation of the body is just, there's an emergency or there's not an emergency. But it isn't true. It wouldn't be efficient. So there's a continual regulation of fuel, of supplies of all of the things we need: oxygen, water, salt, nutrients are continuously regulated according to the need. And what's also regulated is our anticipation of what we're going to need. So we don't wait for the body to cool off and then say, oh my gosh, we have to burn more fat to warm ourselves up. We remembered, oh, we'll take a sweater. And when we get cold, we'll put on the sweater. It saves a lot of fuel. And if you make a mistake and go high in the mountains where without a sweater, you're risking hypothermia and death. So the idea of allostasis is, allo means other. And so the idea is, rather than holding things constant, it's allowing variation to occur to preserve stability. And it profoundly involves the brain. So in medical schools across the US, people teach about the body, and then they teach a course in the brain, but they don't really get them together very much. And our formulation is that the brain is, from the beginning, profoundly engaged. In fact, the whole point of the brain is to regulate the body efficiently. And this started with the first multicellular or bilateral organisms, as I maybe will mention later. A worm, a marine worm, had the first brain, and the point of it was to steer the worm to where there was good stuff to eat and where the right temperature and the right pH, and find mates and stuff like that. And it developed these systems for continual monitoring and regulation. and we preserve them actually. So yeah, so that's the idea of allostasis, is a continuous regulation, and it's really what Toyota invented for the assembly line. Instead of having great stores of materials and too much storage space and running out of stuff, its slogan of Toyota became just enough, just in time. We'll supply just what you need, just when you need it. And basically, it's a sort of allostasis.
Mm-hmm. So to try to put things into an evolutionary context, when it comes to human evolution, in your book, What is Health, you talk about 4 epochs of design that characterize it. Could you tell us about them?
Sure. Well, of course, it's an arbitrary designation, but I found it useful. I mean, I didn't start with this idea. I just, as I was reading and thinking about it, it sort of came to me that, of course, life on Earth began 4 billion years ago. And the first organisms were bacteria and related organisms, and they were really, really tiny. And the source of energy that they had was they had these little power plants that would take energy from the environment, convert it into chemical energy inside the cell, and they were placed in the cell membrane. And so so it was enough to develop chemical reactions to, say, break down glucose, make new proteins, and stuff like that. And so for 2 billion years, bacteria and these other organisms, archaebacteria, there's a variety of classifications, developed their chemical reactions to run this metabolism to a very high efficiency. So they developed the ways to break down glucose into, which is 6 carbons, into single carbons, how to oxidize it to capture the energy. And these reactions really became optimized. Many of them are the sequence is as efficient as you can get. The energy capture is as efficient as you can get. For example, a bacterial macromolecule called ATP synthase takes 2 molecules, forces them together to make ATP, which is our energy currency to drive most of our reactions. The energy it takes from glucose and produces in ATP is 90% efficient. So it's nearly as good as you can make. So the bacteria optimized our fundamental biochemical metabolism, and we retain that. So in some sense, we retain the evolutionary products of bacteria in all our cells, because we couldn't improve them. We could make them worse, but these are just as good as you can get. So we conserve them. So after 2 billion years, there are these tiny bacteria, and they sort of had reached their limit of what you could do at that size scale. And they couldn't get bigger for the reason that if you increase the diameter of a cell, its volume of its cytoplasm increases as the cube of the diameter. But the energy source is in the membrane of the cell, and it only increases as the square. And so there's a geometrical, physical limit to how you can enlarge a cell only with the power plant in the membrane. So what happened? Well, one kind of bacterium captured another kind of bacterium, and we don't know how it happened, but before too long, the power plant of the one bacterium was inside the cell, and it stayed there, and it could multiply. And so now you had a cell that had its power plant in the cytoplasm, and it could keep pace with the growth of the volume of the cell. And so it became a permanent structure of the cell, an organelle, which is called a mitochondrion. So once this bacteria had captured this organelle and made mitochondria, it had essentially an infinite energy source, and it began adding genes, more new genes. It added new organelles, for example, a cell nucleus to surround the DNA and to regulate its expression. And so these cells grew a billion-fold in volume. These are called eukaryotic cells, that are cells with a nucleus, and they can be a billion-fold, let's see, I think so, yeah, a billion-fold larger than a bacterium. And so they diversified, they made all of these different efficient intracellular organelles and became very efficient. And we retain these. I mean, our cell physiology, cell structure is based on what was evolved by these eukaryotic cells between 2 billion and 500 million years ago. So in that sense, we are 2 billion-year-old bacteria. We are, somewhat newer eukaryotic cells. And then what happened with the eukaryotic cells? Well, they're still too small to get very far swimming in water. If on the size scale, water is very viscous. And so for a eukaryotic cell, for example, a paramecium, a single cell protozoan, swimming with all its cilia is like us swimming through molasses. It's a very laborious, energy-producing, slow, and not very effective process. So what happened? Well, the answer to that was for protozoans, some eukaryotic cells to begin to stick together to make a multicellular organism. And then they could drift on the ocean current or make progress by various swimming mechanisms like jellyfish do. It's a slow business, but they do get long distances. And so that generated a whole, period of evolution of multicellular organisms. And it wasn't, so the thing is, you start out, it takes 2 billion years to get bacteria really in good shape, then another 500 million to get eukaryotic cells. But then once you get multicellular animals, things proceeded rather quickly. And by 500 million years ago, we had these marine worms that were bilateral and had a brain. And so once you had that, they produced the arthropods, the insects, crustaceans. They produced vertebrates, and once you had vertebrates, we went through from fish to amphibians to reptiles to mammals rather quickly. And so the first worm was 600 million years ago, maybe 500. The first mammal was only 200 million years. So from 200 million years ago to primates. Primates are, maybe, 100 million years. It was very short. And then from primates to Homo sapiens, or the ancestors that we share, say, with chimpanzees, was only 20 million years ago. So, and then the human species is only about 200,000 years old. So it became a very rapid thing because we had all the parts and we just reassembled them in different ways. So those are the major. Oh yeah, I forgot one epoch. Yeah, so we went three epochs: bacteria, protozoans. Sorry, prokaryotes, eukaryotes, multicellular animals, and then once we got to reptiles, there was another important step to humans, which was to mammals. And the thing about mammals is that they are animals that have they raise their temperature. They can maintain a temperature that's quite a bit above the environmental temperature. And the reason that's important is because chemical reactions are very sensitive to temperature. So there's what's called a Q10 for every 10 degrees centigrade a temperature rises, the reaction rates double or triple. And so these new organisms, once they could raise their temperature that much and hold it there, they could see faster, their brains worked faster, they could think faster, and they could move faster. And so, and they sort of developed as some off branch of a reptile. And so once they could raise their temperature, maintain it this way, they were eating the reptiles and outcompeting them. So there's one other part to this, is that if you're born as a tiny mammal and you have to maintain your body temperature, it requires tremendous amount of fuel. And the most convenient way to supply that fuel was to have the mother give the infant mammal milk. And so lactation became an adaptation that really went very closely hand in glove with what's called endothermy, maintaining your temperature. And to give an idea of how costly that is, a female mouse, when she has 6 little mouselets, baby mice, is feeding them, her metabolism has to be as high as that of somebody who's competing in the Tour de France. They eat as much as they can to convert as much as they can to energy, and so it's a very demanding metabolic state. But that was, I think, the fourth epoch, is the conversion to mammals with high temperature and lactation.
Mm-hmm. So there's another idea that you talk about in the book related to design, that is optimality. And I guess that many biologists out there will not be fond of it, but could you explain it?
Sure. The idea, there are many biologists who, if you say this is optimal or that's optimal, this was perfected, they laugh at you and they call you, I've been accused of this many times, as being Panglossian. Pangloss was a character in Voltaire's Candide, he was a doctor, and whatever bad things happened to Candide or his girlfriend, Pangloss said, well, it's the best of all possible worlds. God created the world, so it must be the best of all possible worlds. Everything will be fine. So I'm often have been accused of being a Panglossian, but the point is, and I'm not alone in this, that there are many aspects of biology that, if you study them closely, you can see that they are as good as the physics and chemistry will allow. And they can't get any better, because, a protozoan can swim as fast as is allowed by the viscosity of the medium. Okay, so it rowing, whatever it can be for that size, it can't get any better. And so, for example, an enzyme, which is a protein, enzymes are proteins that facilitate chemical reactions, and the faster they can work, the faster the chemical reaction, the more product can be produced. But many enzymes, not all, will reach a limit called the diffusion limit, and that is that they react as fast as the two reactants, chemical reactions, can diffuse into the catalytic center of this enzyme, have the reaction, and then diffuse away, and then a new set of reactants diffuse in. That's set by the physics of aqueous diffusion. And there are certain enzymes that actually reach that level. And so they are optimal for speed, and they can't be improved. You could make them worse by tinkering with them, but you can't make them better because they're diffusion limited. Not all enzymes reach that level. for other reasons, because speed isn't the only thing that you want. You may, depending if you have a high concentration of reaction, you might want to reduce the affinity of the enzyme. So there are many in optimizing. Some things are purely optimal. Others are optimized by making compromises between one demand and another. You can't. Some speed and isn't always the only matters. So as I mentioned, ATP synthase, which is at the core of our metabolism, reaches an efficiency of 90%. That's about, that's essentially optimal. You can't improve it. And human designed power plants don't get anywhere near that. So in both of my books, one with Simon and my other, there are many more examples of this issue. And other people have written books. McNeil Alexander has written a book called Animal Optima, and he's got case after case of this. So.
Mm-hmm. So before we move on to issues regarding medicine and modern medicine, just one question that is a little bit different from the ones we've been exploring until now. How can we explain from an evolutionary standpoint things like music, art, play, comedy, and so on in humans?
Yeah, that's a great question. I want to add one more thing to my previous point. One reason that I emphasize the prevalence of optimization and optimality in biological design, human design, is that if we realize how finely things have been tuned over 4 billion years, we might think twice about giving people all of these drugs to make them better, because they're interfering basically with optimized mechanisms. And so you might make things better, but you could make them worse. And when you give people, when you have a medicine that's built on 4 or 5 different kinds of drugs, you're really coming up against this 4 billion years of optimality. Mm-hmm. Okay, so how do we explain music, art, play, comedy, laughter, and all of these other behaviors that have to do with emotion? So I think it's a very important question, because our brain invests huge resources in all of these activities. And yet these activities really belong to so-called leisure time, quote unquote leisure time, and they yield no calories at all. I mean, unless you're a highly paid artist or something like that. But for most people, these activities are not about finding food and making food. It's a cost. So here's my take on this, my answer. Human intelligence is basically cooperative. And the way human societies become so brilliant is that individual human beings with a brain that's only 1.5 liters, once your brain is grown, it's inside your skull, it can't get any bigger, and it can't add more computation, because there's again a physical limit. But what I think happened in human evolution is that by design, every brain is different. So when we have maybe 200 cortical areas that are distinct, but they're not the same in you and me. Some people, some very brilliant people, are unable to recognize a face. And Oliver Sacks was a good example. He wrote about people not being able to recognize faces, but he had this in himself. But he was extremely musical, and he was extremely empathic, and he had all kinds of other abilities. Extremely good writer, but probably the areas in his cortex that are just are specialized for recognizing faces and remembering them. Either he didn't have them or they were diminished in size and so on. There are people who are born with musical talents. I wasn't one of them. And so they pick up an instrument and start playing it really with very little instruction and so on. My son is sort of like that. I mean, he wasn't Mozart, but definitely I noticed early that he had a talent that I just didn't have. Same for art. The cave paintings that decorate the cover of my book, What is Health, are made. I mean, if you look at these cave paintings in Europe or in Indonesia, where they came 45,000 years ago, they're not paintings by your average amateur. Cartoons show cavemen making these stick figures. These are artists, serious artists, and they must have been supported to do that work by people in the community who fed them while they did their art. They're incredibly, the fact that we still respond to them, paintings that were made 45,000 years ago, means that somehow we're the same people, and we can get this art. So once you give brains these different talents, innate talents, people practice them. You practice what you're good at because that's rewarding, and we can discuss why that is. But when you practice what you practice, you become. And so we generate a community of hunters, healers, navigators, artists, musicians, and so on. And so, but what's the point? Why do we do that? Was the community support musicians and artists? Well, because we also, by this specialization, became individually sort of lonely in a way. I guess you would call it. We have built-in existential loneliness and sources of interpersonal conflict. And so, but if you're going to cooperate in a community to share this communal intelligence and exploit it, you have to get along. And so I think that I try to document in my book that the reason our brains invest so much as a community in art is that it relieves tensions. Sorry.
No problem.
It relieves tensions and it helps people get along and reap and calm down and reap the benefits of this communal intelligence.
Mm-hmm.
So I think that's it.
Yeah. So we will get now into modern medicine, looking at the time. Perhaps we will explore 1, 2, or 3 questions, and then if you would like, I would suggest for us to schedule a second interview to get into the rest of the topics more in depth, because there are still many questions I have here, but we don't have enough time today. But how do we distinguish between a disorder and merely expected variation?
Mm-hmm. Well, I put this a little differently. I see that all processes vary, and that's the point of allostasis. All processes need to vary according to the context. And every variation in our physiology has some cause. Modern medicine, conventional medicine, often measures these variations. Before I see my doctor in the US, I get a series of blood tests, and they look at the test. They see, well, this is all in line. This is out of line. We have to fix this, and so on. And this is not entirely ridiculous. I don't want to scoff. It's the intelligent aspects of modern medicine, but it sees many variations as it calls them a disorder or a dysregulation. But often these variations are not a disorder or a dysregulation. They are what you would predict if you understand the context of what the individual is, of the individual's existence. Often there's nothing broken, but there is maybe something wrong. And so if you take some variation in a human's functioning for a long time and you push it beyond where the design was ever intended for, then things can go badly. And so let me choose two examples. One, of course, is blood pressure. I point out that in hunter-gatherer societies to this day, there's still some that exist. I have visited some of them myself, or horticultural societies. People's blood pressure does not rise with age. It's around 100, a little over 100, over 70 or something like that, and it's that way forever. And they are basically lean. They have a lot of exercise, and they follow their own daily rhythms, but their blood pressure is low. In the U.S., in industrial countries, when children go to school, their blood pressure at like age 7 starts to rise, and it rises so that by graduation, 18 or so, maybe a quarter of the population already have pressures in the hypertensive range. Nothing's broken. It's just that the neural mechanisms that respond to various sorts of challenge are turned on a lot. And so the blood pressure goes up, but instead of coming down later, it doesn't. And so when you have chronic elevation of temperature, the cardiovascular system adapts. What happens? Your arteries thicken. It's like your biceps when you're lifting weights gradually adapt by thickening, because they predict, oh yes, we're going to have higher demand. We will make more muscle. So that's what happens in the smooth muscles of arteries. They expand, sometimes even beginning to occlude the lumen of the vessel. And so it takes more pressure to drive the blood just through normally. And that's a sort of addiction where what you started seems reasonable, but now you rely on it. And eventually, if you have hypertension, it causes inflammation, it causes all kinds of other problems that lead eventually to stroke, heart attack, kidney problems, and so on. And so even though it's not, doesn't start out as a disorder, it can eventually lead to death and disability in a lot of ways. And it's a major cause of death and disability. But medicine, in medical schools, if they call this essential hypertension. And the word essential in medicine means we don't know why it is. But I think I know why it is. I think it's pretty clear. It's because we live pretty much as a society at a high level of social tension, really. And an example of this is in the US, the communities with the greatest level of hypertension, the greatest level of the problems from hypertension is the Black community, the African American community. And in the US, the reason is that in the US, Black lives still don't matter. And so that they are under constant pressure and threat, for example, of police, as we've seen over the last few years. And so they have the worst hypertension. And in medical schools, they tend to blame it on genetics, which doesn't turn out to be true, because their genes are the same as the genes for West Africans and who emigrated, were brought here as slaves, and as Caribbean Blacks. But those people with the West African, Caribbean Blacks have a much lower levels of hypertension than the US. So that's one example. Another, are you with me? Are we doing all right?
Mm-hmm. Yes.
Uh, Another example, which is highly relevant, is type 2 diabetes. It's a major contributor to cardiovascular disease. It also is a contributor to high blood pressure. And it's all kinds of other problems. And modern medicine calls this, again, a regulatory disorder, dysregulation of carbohydrate metabolism. But and it's very complicated, but the short answer is if you eat high levels of carbohydrates, sweets, and sugar all the time, every time your blood pressure, your blood sugar goes up, it evokes insulin to be secreted to reduce it, to regulate it. But what happens when you have high insulin, called insulinemia, high insulin blood levels of insulin, is that the cells that have insulin receptors, that are protein molecules that bind the insulin and then respond to it, they reduce their sensitivity. And this is the same thing is true for all biochemistry. If you have receptors and you challenge them with high levels of the chemical, they reduce their sensitivity. And so actually the number of receptors on a cell, insulin receptors, is reduced. The cell gobbles them up and reduces the number. So modern medicine calls this insulin resistance, as though somehow, the cells were incomprehensibly resisting the chemical that they need. But it's a misnomer. I mean, it's a confusing term because it conceals the fact that this is just normal adaptation. And it's a complex adaptation. There are people who are studying this all over, very complex adaptation. But nevertheless, insulin resistance is a response, is a predictable response to chronically high levels of glucose. And you can show it in a fruit fly. If you feed a fruit fly on a high carbohydrate, purely on a high carbohydrate, it will develop essentially type 2 diabetes, and of course, then we get to mental disorders and whether those are actually disorders or just normal variation that becomes a little more extreme in some people. The circuits that we need for normal functioning, if you get out on the tail, maybe are a little more sensitive in some people to stress and abuse and so on. And people can go mad, but it's sort of it's not I think an intrinsic disease, or we don't know of any circuits that are broken really. So anyway, I'll stop with that.
Mm-hmm. Okay, so do you think that the fact that modern medicine is based on the principle that the body is self-regulating, I mean, on the principle of homeostasis, that's a problem?
Yeah, I think it's a problem because it teaches physicians from day one that they should examine the body and all its processes independently of the brain and the mind and emotions. And so, it's incorrect. It's a very distorted view of the body, and that's really my objection to the idea of homeostasis, and because it just cuts off the brain as a constant regulator for reasons of efficiency, and people, people give drugs to change your chemistry. I can change your chemistry by shaking your hand or smiling at you or patting you on the shoulder. So the idea of coining allostasis was that if you want to get past homeostasis, you need another word to refer to it. And so I've been selling this word for 35 years, or almost 35 years. And last week, I noticed there's going to be a meeting sponsored by the Federation of American Societies for Biology, FASEB, next summer in New Orleans, and it's co-sponsored by the Endocrinological Society. It's on allostasis. And so I'm delighted. I think it's a term that's, it's not on everybody's lips, but it's gaining a little bit of respectability.
Mm-hmm. So, Dr. Sterling, I think this would be a good point to finish our first talk, because then we will have to get into other issues surrounding human evolution and human health in modern industrialized societies. And so lots of issues to explore next time, if you agree to schedule a second talk. And just to tell again that the book is What is Health, Allostasis and the Evolution of Human Design. Would you like to mention any places on the internet where people can find your work?
Sure. I do Twitter, actually, and my Twitter name is What is Health 21. At What is Health 21. You can find me there. Yeah. So thank you. I enjoyed this, Ricardo, and I'll be ready when you are.
Hi guys, thank you for watching the interview until the end. If you like what I'm doing, please consider supporting the show. It's thanks to people like you that it keeps running. I will leave links in the description box to Patreon and PayPal. Any amount, even just $1 per month, would already be a great help. Otherwise, and if you like what I'm doing, please share the interview, leave a like and hit the subscription button. This show is brought to you by Enlights, learning and development done differently. Check their website at enlights.com. I would also like to give a huge thank you to my main patrons and PayPal supporters: Karen Litska, Anne Blanchett, Pererga Larsen, Lau Guerrero, Francis Ford, Hans Frederick Sunder, Ricardo Vladimiro, Craig Healy, Adam Kessel, Olaf Alex, Jonathan Wiesel, Jacob Klinkby, Matthew Whitting, Bernardo Wolf, Tim Hollacy, Enrique Alenia, John Connors, Paulina Barron, Philip Force Connolly, Jerry Mueller, Herbert Gintis, Rutger Voss, Bo Weingard, Rebecca Newberger Goldstein, Dan Demetriou, Robert Windiger, Rui Inácio, Arthur Coe, Zup, Marco Neves, Colin Holbrook, Susan Pinker, Bernardo Seixas, Pablo Santurbano, Simon Colombo, Jorge Espinha, Phil Kavanagh, Corey Clark, Mark Blythe, Roberto Inguanzo, Michael Stormir, Eric Neumann, Samuel Andreff, Tiago Nunes, Bernard Ugni, Alexander Dunbauer, Fergal Cusson, Evan Bodrenko, Al Herzog, Don Ross, Jonathan Librant, Oslan Bullitt, Nathan Nguyen, Stanton T, Samuel Correa, Eric Hines, Mark Smith, J.W., João Eira, Tom Hummel, David Sloan Wilson, Yacila Des Araújo, Eden Solon, Romain Roach, Dmitry Grigoriev, Diego Londonio Correa, Tom Roth, Yannick Punter, Adana Rusmani, Charlotte Bliss, Nicole Barbaro, Adam Hunt, Pavel Ostazewski, Nelek Bach, Catherine and Patrick Tobin, Al Ortiz, Guy Madison, Gary G. Hellman, João Linhares, Lida Cosmidi, Sam Afzal, Adrian Yegi, Nick Golden, Paulo Tolentino, João Barbosa, Jules Price, Edward Hall, Edin Bronner, Franca Bortolotti, Gabriel Pons Cortez, Ursula Litske, my producers: Izar Webb, Jim Frank, Lucas Staffini, Akion Gilligan, Luís Caetano, Tom Vanagdam, Curtis Dixon, João Linhares, Benedict Mueller, Vega Guidi, Sardas France, and Thomas Trumbull, and my executive producers: Michel Rugieski, Rosie, James Pratt, Matthew Lavender, Sergio Quadriano, and Bogdan Kanivets. Thank you for all.